is the importance of the cell membrane? So write down your essential question under the title. What is the importance of the cell membrane? Page 89, your title, cell membrane, and your essential question. All right, ladies, you have your essential question right now. Let's try 89. All right. So today we're covering the cell membrane and why it's important. So we went over homeostasis, which means balance, right? We need a stable internal environment. So the cell membrane is like a barrier in the cell between the internal and external environment. So for example, water balance. It regulates the entry and exit level of water through osmosis. So there's an example that Ms. Copin was giving earlier at a frat at a college. There's in order for you to go in to the frat, they made a student, I think back in 2021, have 20 liters of water in 1 hour. So what do you think happened?
He died.
Yes. But what do you think happened? All that water. Why so much water? Your body system, right? Okay, so that's what happens. Through whenever it went through osmosis into the cells swelled up. No more water is able to go through. So there's a reduction of sodium. So even whenever you're exercising or you're in sports, right? You shouldn't drink a lot of water in a little bit of time. A professor once told me if you're drinking a gallon of water, even to add a little bit of salt, not that tastes like salt water, but even a little bit, it's good. Because you're drinking so much water, you're supposed to have a little bit of salt too. If you drink so much water, it reduces the sodium in your body and you can die. So everything in excessive is bad for you. So that's the cell membrane. It sees like what water goes through, and also that's osmosis, and you will start seeing that we're having a lab on osmosis and diffusion. Like everything that comes in through food and it breaks down through nutrients. So that's what we're learning today, okay? So we are going to watch a video first on the cell membrane. We're gonna take some notes, and then we're gonna get ready, set up for an osmosis lab. So we are gonna be in the lab probably tomorrow. So it's gonna be a fun day. So let's watch the video. All right, here's the video on the cell membrane.
In my second year of teaching, I was looking to improve an osmosis as I had done the year previously. Osmosis, if you remember from our osmosis video, involves water traveling through a semi-permeable membrane, potentially a cell membrane. And I wanted a cool way that students could model this in different scenarios. And one of my colleagues told me about this egg lab. I won't get into the whole lab, but it was actually one of the first steps of the procedure that got to me. To get ready for the lab, my colleague told me, you can soak eggs in vinegar for 24 to 48 hours and the shell comes off. Oh, so I need to make hard-boiled eggs. No, raw eggs. But if the shell comes off, well, that's the whole point. What's underneath the shell is going to mimic a cell membrane. It's kind of modeling how a cell membrane would function. You know, if the whole egg was actually like a cell. So then you run different scenarios with it in your osmosis lab because it will be a semi-permeable membrane like a cell membrane. Very important when you're fighting pathogens. They can also be involved in many kinds of cell signaling. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against many viral and bacterial diseases. Well, that's it for the Amoeba Sisters. Let me remind you to stay curious.
Alright, so I passed out your doodle notes. We're gonna go over them. So, this is the cell membrane. There's different components within the cell membrane. As you see, there's different colors. Your paper's blank, but you will get to color it at the end. So this is the external environment on the blue. It'll be the outside of the cell, and the pink is the inside of the cell. So I'm gonna go ahead and label each box. Some of them will be empty, and then I'll tell you which number to label as we go. All right, so for number 4, it would be this right here. It's called an integral protein. So you will label it in the box, and I'll write it over here on the side. Number 5. These right here that are in green are called receptor. So that's a receptor. The little blue proteins at the end are called receptor protein. And this is what you call, number 7, a phospholipid bilayer. So right here on the side. Number 7. Number 8, right here at the bottom, inside the cell, is called the surface protein. This right here would be a channel protein. Number 9. Number 10 would be a carrier 11, peripheral protein. And number 12, it's cholesterol. Cholesterol. So at the bottom left where it says cholesterol, where there's a little cholesterol and a phospholipid, it says keep your distance. In the little bubble, keep your distance. And it says, it's fill in the blank, so cholesterol, this is a type of steroid. It regulates the fluidity of the cell membrane. At low temperatures, it keeps the phospholipids spread out. So fill in the blank. Cholesterol, it's a type of steroid. It regulates the fluidity of the cell membrane. At low temperatures, it keeps the phospholipids spread out. Alright, where it says phospholipids in the middle, the little blue circle is the head, and the little two lines would be the tail. So the head, where it's a little mark, write down hydrophilic. That's the head, and it is hydrophilic. Hydro meaning water, philic meaning loving, so it loves water. So the head loves water, so hydrophilic phosphate. Where it has the tails, it's hydrophobic, so the tail is hydrophobic. Doesn't love water, so fatty acid bearing. And you can make a note on your doodle notes that hydro means water. So that should fit in the middle portion right here.
The tails have a phobia of water. Do you guys know what a phobia is? No?
They're scared?
The movie
They're scared?
Arachnophobia? What was that? That person afraid of?
Spiders.
Spiders. Okay.
Alright. Okay, so the last portion, the fluid mosaic model. So the phospholipids are able to move around each other like a fluid. They have different kinds of proteins and molecules embedded within the bilayer. Like titles in a mosaic. So that's the last portion of the note, sorry. Alright, so you won't be able to color right now because we are setting up for our osmosis lab. Alright, so Miss Joaquina is passing a Sharpie to each table. Miss Joaquina is passing a Sharpie to each table. You have 3 cups. We are setting up for our osmosis lab. So in each cup, you're going to write something. So please pay attention, that way you know what to write. So first of all, in each cup, you are gonna put the period we're at. So we are on period 4, dash the table that you're at. So this table, you guys will be 4-7, 4-5. You're gonna write down 4-4 on each cup with the Sharpie, 4-6 on each cup. Nina's table, 4-3. You guys are 4-2, 4-1. Alright, so each cup you will have the period and your table number. That way we know that is your cup.
Can you hold your cup so you can listen? We're not flipping the cup right now. Just put the cup down. Thank you.
And take care of your cups, okay? Because that's what you're going to use the lab for. Okay, so each cup, so it's gonna be cube 1, cube 2, cube 3. So first cup that you have with your Sharpie, you're gonna label it cube 1 tap water. This is what you're gonna write after you label the period and the table you're at. Cube 1 tap water with your Sharpie. The second cup, you're gonna label it cube 2 salt water. The third cup, you're gonna label it cube 3 distilled water. Alright, so start labeling each cup. Each cup should have your period and table number, and each cup will label cube 1, cube 2, cube 3.
Oh, girl. All you see is right there. Stop. I made a joke out of it. Out of my. All you see is right there.
And then
So background is me. Clearly. That's it.
Oh.
Did the body not? Is the body not body? Oh.
Why'd you give me that?
Because you're lazy. Don't worry, I'll keep your secret safe, guys.
Cube 2.
cube 1, 2, cube 3 on each cup. Write down.
This would be better if it was a kid. I have a picture of me.
You want to be able to see cube 1 tap water on your cup.
Stop flipping the cup. I can ask you again. No, it was you. I'm looking right at you.
Oh, that smells right. Guys, I think I need to remind you what you said to me the other day.
Okay, we're gonna pass Deco cubes. Deco. We're gonna pass out Deco cubes. So now that you have,
Do it. Yes, sir.
did everybody get to label your cup?
Is it white? Oh, they look like sugar cubes.
It's a cute, isn't it?
Oh, ladies, that's not nice. Oh, it is so cute. It's so
So as you're getting the Deco cubes, you are measuring them. So right here in the first box of your osmosis lab, we're preparing for your lab. So it's the initial volume. So for the cube one, each one should be in a different cup, in a different cube. Okay? So each one of your groups, you guys can take turns. There's more than 3 people in your group, so each one of you will measure. Alright, you guys listen over here. So you're going to measure the first one that goes on cube 1. One of you can measure 1, another person can measure the second one, and another person can measure the third. So what you're gonna do is you're gonna take the length. Okay, this is gonna be in millimeters. Length times width times height, and that gives you your volume. If you want, you can do the math on page 90, but on your paper, we only need the total right here. Alright, so the length times width times height, so this is your,
stick to my
the centimeter side, you will be using the centimeter side, and you're counting each millimeter. That will go in cube number 1, cube number 2, cube number 3. So everyone should be measuring right now. If you're in a group of 3, you should all be measuring 1. Yeah. Where's the other one?
Just tell them a female.
These are centimeters. How do you measure? Oh, wait, I see those?
Yeah.
There's no way.
See the little line?
Very little line, yeah.
You're gonna use those two. I got 8. Put it at 0.
I think mine counts.
Do I count 0 or no?
No.
Okay, that's good.
Yeah, mine said one.
You want me to tell you mine?
No.
What are you talking about? 6 centimeters?
I don't know how to measure.
Those are important.
Wait, what do we do? After we measure?
I was never gonna measure. I did not pass that.
I got 6, bro.
So you're
Must have been
measuring
dumb.
all the sizes and then the height, the length, and the width.
Mine was smaller. I'll name it Tiffany. Tiffany.
So like,
That usually weighs.
yeah, like right here.
Mine's 7, actually. Oh, we write down this. Initial volume. Wait. Oh, we also gotta get width.
What is a
Well,
width?
we just measured this the width.
What is a width?
So we have like length. So the height would be length, width.
That don't make no sense.
So you're
One side is length.
Oh,
One
you
side is
also measuring.
Oh, and you gotta do height.
Don't forget the
Girl.
Like, look right here. You see how it has length and width?
So why do we have to do this? I'm like, I even need science.
the total
Paper
right
and
here.
draw.
For whichever.
We'll just count.
So that would be.
I thought you were doing that to me. I got scared.
It's like a centimeter tall.
Seventh of the 17th.
It's 7 and
What's a cube? So 7 times 7.
oh, bro, this cube is not even at all.
Oh, my goodness.
It's like sticky to the paper.
I'm gonna say 7.
I'm gonna say. I got 7 sevens. I got 7 something and then 6.
Hey, what's the width?
How do you do it?
Yeah.
I have length and I have height.
So you measure one side. So no matter what, this thing is 7 again. Because like
How much is it? 7, 7, 7, 7, 7, 7.
I have 7, 7, 6. Times all that.
Oh yeah, dude, we have to do length times width times height.
So 7 times
I got
7
1
is
centimeter.
whatever, that comes out to 6.
What the freak? Why is your cube so big?
I have 1 centimeter, right?
They are not edible.
Wait, do I have to do one at a time? Or do I have to do it all at once? Like, do I do 7 times 7 and then have that twice?
Okay, my jump is like 8
Your jump?
millimeters.
Yeah.
Millimeters.
Why'd you do my jump?
I meant these, bro. You weird, bro.
So this is cube 1.
Do you think a fart? Do you think a shark farts?
Oh.
So whatever number you got, write down millimeters because you're doing this in millimeters.
I'm gonna look this up, guys. This is something that I need to do.
The what?
How do you find the width?
Okay.
So let's say.
So you're doing it like this.
What's the length? Isn't it that?
Pero de little bit of faded in like that. You're gonna do this. That's what you're doing. And you're gonna put it up. You're gonna do this.